Elearning Jamaica Limited, in association with the Ministry of Education and Czec Jamaica Limited, present a series of e-learning videos geared towards preparing students for CXC examinations. This series focuses on biology. There are 10 videos, each lasting 20 minutes. Your narrator, Michael Anthony Cuff.
Welcome. This presentation covers Section E of your biology syllabus, which deals with the environment and human activities. At the end of this, you will have a better understanding of the term ecology, how organisms are dependent on each other and their surroundings. Also, how the features of both a terrestrial and an aquatic area impacts on living things. When you look around you, the surroundings at your school, home, and of any area is called the environment. Different organisms live within the environment, and where these organisms live is called the habitat. Let us look at a farm, for example. It is the habitat for many organisms. However, its inhabitants provides a good example of the interrelationships to be discovered. What do you think are some of the features to be found on the farm? The farm will have a special type of soil, one that is very fertile in order to enhance growth of plants. There will be a variety of animals like cows, goats, and chickens. A variety of plants such as a special type of soil with rocks, minerals, and the remains of dead organisms. Bacteria and other microorganisms will also be present. Let us put all those pieces together. Each component works together, thereby forming a system, more specifically, though, an ecosystem. Look carefully at the ecosystem being depicted. What two categories can the components be divided into? If you said the living or non-living, you are correct. An ecosystem is therefore divided into two categories: the living component, also called the biotic component, and the non-living component, also called the physical or abiotic component.
In this section, you will be learning about the different methods used to establish the number of individuals within a population or community, and how to choose appropriate methods for obtaining population numbers. Communities and populations are generally large and not evenly distributed, making it tedious to observe each individual. Therefore, in order to estimate the numbers of a population within a community, one has to obtain samples of individuals of each group. The quadrat. The quadrat is a square generally made of a wooden or metal frame of a specific area, usually 0.25 meters squared or 1 meter squared. The quadrat is used for sampling vegetation, slow-moving animals, as well as organisms present on the surface of rocks in a stream or on a seashore. The quadrat may be used for random or systematic sampling. Sampling may either be random or systematic. From the samples collected, one is able to estimate the number of organisms within a given area of ground or volume of water. This is the species distribution. Species distribution involves the species density and species frequency. Species density is the average number of individuals of a particular species within a given area. Species frequency is the percentage of sample units in which a species occurs. Samples may be obtained by using quadrats, nets, bottles, and transects. The net. Nets are used for sampling small moving animals, and it is generally made of a small mesh with a handle approximately 1 meter long. The mouth of the net is swept through the vegetation in order for dislodged animals to drop into the mesh or bag. The size of the mesh used will determine the size of animals captured. Bottles. Bottles, which are of a known volume, may be used to sample small aquatic or terrestrial species. In an aquatic area, samples of either pond or river water can be collected to be analyzed with the use of a hand lens or microscope. On land, bottles are used for setting pitfall traps. The bottles are often sunk randomly into the earth below ground level, containing food to attract animals. The opening of the bottle may be covered with leaves and sticks or with a stone lifted off the edge by other stones. The trap catches arthropods such as centipedes and beetles, which are nocturnal. Transects. Using a transect requires sampling along a straight line. The line transect is useful for sampling habitats where there is a clear change in conditions and distribution of vegetation or animals from one point to another. A length of cord with marked intervals is stretched over a habitat, and any plant touching the cord is recorded. This may be done for the entire length of the cord or at regular intervals. The quadrat may be used to calculate species density and species frequency. Let us see how. Look on the table on the screen. Species density is obtained by averaging the number of individuals of a species in each randomly thrown quadrat in a given area. This example uses a quadrat size of 1 meter squared. In 10 throws, a total of 30 plants were counted. The average of plants would be 30 divided by the 10 throws. The species density would be 3. The species frequency, however, would be the number of times the species is present in the total number of throws. For example, from the sample, the species was present in 7 out of 10 quadrats thrown randomly. The species frequency is therefore 7 out of 10 multiplied by 100, equaling 70%. Terrestrial and aquatic environments will have certain features in common namely air, animals, plants, and a specific soil type. Air comprises oxygen, carbon dioxide, and nitrogen, which are gases that are used by both plants and animals on a daily basis. The oxygen is relevant for the process of respiration, and carbon dioxide is necessary for photosynthesis, and thereby lead to the formation of carbon compounds within plants, animals, and the environment. Nitrogen, however, is not in a usable form and must be converted to a nitrate in order to be used. This is aided by nitrogen-fixing bacteria or lightning. The nitrates will enter plants and animals. However, it may be returned to the soil by nitrifying bacteria and returned to the air by denitrifying bacteria. Soil is a natural resource that is a product of the interaction of several factors such as parent material, time, climate, plants and animals, and the degree of slope. Soil is made up of both organic and inorganic materials. The organic materials are plants and animals that are or were once alive, and inorganic materials are mainly rocks and mineral particles. This student wanted to identify the components of soil and therefore conducted the sedimentation method of examining soils. Some soil from the school garden was placed into a large cylinder to which he added some water and observed. The components of the soil settled out into different layers. This was due to size and weight of the soil particles. Can you see the components of the soil? They are humus, clay, silt, sand, and gravel. Clay, silt, sand, and gravel are inorganic, whereas the humus is organic. Humus is a dark, sticky organic material formed from the decay of various organisms. Humus is rich in nutrients that plants need. It also holds water in the soil. Soil also contains air, water, and dissolved minerals. There are three main types of soil: sand, clay, and loam. Do you know some of the properties of the different types of soil? Sandy soil has mostly large particles and large air spaces. Therefore, a lot of air is present. Clay soil has mostly small particles, small air spaces, and therefore little air is present. Loam is a mixture of clay and sand particles. Therefore, the air spaces are intermediate in size. Another property is that of the ability of the soil to drain or retain water. This may be tested in the lab. Collect samples of the three soil types. Collect 3 filter funnels and place a folded filter paper into the neck of each. Measure equal volumes of sand, loam, and clay soil. Place a soil sample into each filter paper and rest the filter funnel into the neck of a measuring cylinder. Into each soil sample, gently pour each volume of water. Then, record the volume of water that drained through. To find out the amount of water that was retained, simply subtract the volume of water added to the soil from the amount of water that drained through. For example, if you poured 100 centimeter cubes of water into the soil and it drained 60 centimeter cubes, then the amount it retained would be 100 minus 60 equaling 40 centimeter cubes. Since soil contains these different components, it would provide plants with nutrients, air, and water for growth.
Let's review.
The different methods used to establish the number of individuals within a population or community, and how to choose appropriate methods for obtaining population numbers. Sampling may either be random or systematic. From the samples collected, one is able to estimate the number of organisms within a given area of ground or volume of water. This is the species distribution. Species distribution involves the species density and species frequency. Samples may be obtained by using quadrats, nets, bottles, and transects. Terrestrial and aquatic environments will have certain features in common, namely air, animals, plants, and a specific soil type. Air comprises oxygen, carbon dioxide, and nitrogen, which are gases that are used by both plants and animals on a daily basis. To identify the components of soil, and therefore conducted the sedimentation method of examining soils. There are humus, clay, silt, sand, and gravel. Clay, silt, sand, and gravel are inorganic, whereas the humus is organic.